Can everyone see my screen? Cool. All right. Thank you, Mike, for giving me this exciting opportunity to present my work to your lab. And I'm super excited to hear what y'all think about it and get your feedback on these ideas. Okay, so my background is in computer science and experimental neuroscience. And I've always thought that understanding the brain requires somehow connecting these two fields to a greater degree than what we already have. And so I began my career with the belief that this sort of connection needs to be made at the level of perhaps tens of thousands of neurons. But very soon, as I dived deeper into the biology and I thought more about this problem, I found myself being compelled to think about solutions at the molecular level. And right now, I think that solving cognition at a mechanistic level might lead to some broader implications across many other subfields of biology, not just neuroscience. So what I'm going to do today is I'm going to present a theoretical model that demonstrates that with a handful of very simple editing rules on RNA molecules, you can achieve something called universal computation. Okay, so to properly convey what the motivation and the implications of this model, I'm going to first give you some background that sets the stage for the question that I'm trying to answer. And please feel free to interrupt because I kind of want to make sure that everyone's following all of this. I'd much rather not go through a rehearsed version and have people understand the model as much as possible. Okay, so first, let me start off with the broad topic of biological computation. Okay, so computational problems arise at many different domains of biology. For example, when a bee has to gather nectar for the hive and compute its spatial location and keep track of where it is, or in language acquisition in humans, in human children, or a single cell behavior, or in domains like embryonic development. These are all domains that heavily involve computation. Now, what mediates these kinds of computations? We think that for the domains of cognition and learning, it's done through neurophysiology, neural network dynamics, plasticity rules, and things like that. And for cell behavior and development, we think the computations there are mediated through molecular transduction pathways, gene regulatory networks, non-neural electric signaling, and things like that. But the widely held assumption is that these are the main building blocks of computation, and that biological organisms accomplish these highly complex transformations of information by putting these kinds of building blocks together in the right way. And maybe there's some other building blocks out there that are used to optimize computation in special cases, but these in principle should be sufficient for life's purposes. But the question I want to ask is, are we sure about that? And how do we know that these are sufficient? And I think that there's some fundamental insights from the theory of computation whose implications for our study of natural organisms still haven't fully been explored yet. Okay, so what does theory of computation say? What does computation even mean? How is it formalized? And can we apply it to biology? So in the theory of computation, we can define every problem, every computation problem as a function that maps an input domain to an output domain. Okay, so for example, the problem of addition, we can define the input domain to be pairs of numbers and the output domain to be a single number. Now, a computation system is something that solves not just one, but many computation problems. And the function that it solves is determined by its descriptor, which could be the configurations, the instructions, the parameters, the text that represents the code. So for example, Python, the programming language, is a computation system. The descriptor is the text, the code that you type in, and that determines the problem that it solves. It determines the input-output function. Another example is neural networks. Neural networks are a computation system. The descriptor can be defined as the set of nodes, the connections, the weights, the activation functions, and every descriptor defines a different input-output function. Okay, so what we've learned from the theory of computation is that different systems, different computation systems, have different scopes of problems that they can solve. Okay, so as a simple example, one computation system that we can imagine are Boolean logic gates, or combinatorial logic. So you can put gates together to form these circuits, and then you can ask, okay, what kinds of problems can Boolean logic circuits solve? And crucially, we're talking about circuits that are finite in size. The descriptor is always finite. It should be, you have to be able to convey it in a finite amount of time. So you can use this this problem to solve any problem with a finite size domain. So for example, 8-bit addition. All right, you can create a circuit that performs 8-bit addition, but you can't create a circuit that solves unbounded addition or addition with arbitrary size. However, there are programs, like you can write a program in Python that can solve problems that deal with arbitrary sized inputs. So I can write a code that adds two numbers without a limit, without an intrinsic limit to how long the inputs can get. Okay, so combinatorial logic, the Boolean logic circuits that you're seeing here are a pretty weak system of computation. Usually when people draw the hierarchies, it kind of falls into the innermost layer, and it's thought of as a very weak system. All right, one of the most, I think, most fundamental discoveries, mathematical discoveries of the 20th century is that with a very simple set of rules, you can get to something called universal computation. And what that means is you can create a system that can solve any solvable problem, any solvable computation problem. And the most popularized example of a universal computation system are Turing machines. Turing machines are these machines that work on a one-dimensional tape. In every square of the tape, there could be a symbol, and then you have like a set of instructions that tell it, that define very simple operations for how to interact with a tape. You know, if you read this this symbol, write another symbol, and then go to another line in the code. And again, the length of the program has to be finite. And with this system, you can solve any computable problem, meaning that for every problem, for every computable function, there exists a descriptor that solves that function. Okay, and the crazy thing is it doesn't get more powerful than this. So you can give it a larger alphabet, you can give it a 2D sheet instead of a 1D tape, you can give it fancier instructions, but no matter what you do, you can't take it to a higher power of computation. You can't give it the capability to solve new problems that it couldn't have solved before. Now, there are many other universal computation systems that don't even look like Turing machine. And in fact, it took mathematicians of the 1930s quite some time to realize that the many competing models of computation that don't really look like one another are actually all equivalent. So it's not obvious like a general recursion theory. Yeah, good.
So when you're saying it's not more powerful, you're not saying it's not have no less or more speed. It's just the type of the problem.
Yes, that's a
you can
very
do the
good
parallel. It will be faster, but not solving different problem.
Yes, absolutely. That's a very good point. So you could change the architecture to get improvements in the algorithm complexity, or the memory efficiency, all that kind of stuff. But in terms of the mapping of problems to descriptors, you can't expand the types of problems that it can solve. You can't add a new kind of problem. You might be able to make it faster. Yeah, that's a very good point. Yeah. Okay. So this fundamental insight that you can really easily reach a system of computation that's capable of solving any computable problem. This has led to the birth of computer science. It's revolutionized and shaped modern technology, but what about its implications for biology? Okay. Now, the thing I want to claim is that the kind of systems that we have so far envisioned for computation in biology do not reach this level of computation power. Okay, most of the models of biological computation can be formalized in terms of finite dimensional dynamical systems. Now, there's some work showing that finite dimensional dynamical systems are universal, but the overlooked fact about this is that the kind of networks and dynamical systems that have so far been shown to be capable of universal computation, they lack a certain feature that called structural stability that makes them impossible to build practically or impossible to find to occur in nature. And there's a really nice paper by Chris Moore that explains this and the rationale behind why structural stability is a criteria for practical realizability. Okay, so this opens a problem for us to solve. Why haven't we found a universal computation system or even been able to imagine a universal computation system in nature? And maybe you might say, okay, that's fine. Biology is messy, and it gets by with what it can, and it doesn't really require universal computation for the kinds of tasks, the kinds of computation tasks that it wants to perform. But I find this idea difficult to accept, especially given that it's very easy to accidentally stumble upon universal computation. So these are three examples: Wang tiles, Conway's game of life, Wolfram's Rule 110. And none of these systems were initially intended to serve as a powerful computation system, but they were later proven to be universal. Back in the 1930s, also, the different kinds of computation systems that the mathematicians came up with, general recursion theory, lambda calculus, and Turing's automatic machine, Post's machine, all of these ended up being the same. It seems like there's like a bucket of computation power that's really easy to accidentally fall into, okay? And we use universal computation systems. We use the von Neumann architecture for things that don't really require universal computation. We use them in toasters, vacuum cleaners. We use them in appliances that could have been fine with just analog computers. So the way I think about it is it's easy to reach, and there are many in terms of survival and reproduction, there are many benefits to having a computation system. And also we know that evolution is capable of building highly complex systems like that abide to the principles of optics, like your eye, or mechanics. So why not principles of computation? All right, so I just want to, for the rest of this talk, let's just entertain the idea that there is a natural universal computer, but we just haven't found it yet. What could it look like? All right, and I think the most promising place to search for a universal computation system is at the level of DNA and RNA with polynucleotides. And the reason for this,
Can I ask a question first?
Go ahead. Yeah.
Why do you say that physical computers we find in biology are they cannot be built, they are unstable?
Yeah. Okay. There's this property called structural stability. Sorry, do you hear me? I don't see my. Yeah, okay. There's this property called structural stability, which some dynamical systems have it, some don't. What it means is that the system we're talking about has a non-zero measure in the space of all dynamical systems. Meaning that there's a neighborhood of close enough dynamical systems that are homomorphic to this system, meaning that the number of orbits, the number of like attractor points, all that is the same. You can map the space of nearby dynamical systems to that dynamical system. If a system is not structurally stable, that means you can't approximate it as you get closer to it. It's not like the closer you get, the more it resembles the intended dynamics of the system you're trying to construct. So even if you construct a system that's close to with an error of one part per billion, you still haven't created a system that can be mapped onto the intended system. And here we're talking about noise, not during runtime, but noise in the parameters. So you have to fine tune the parameters of your system so much that there's zero noise. There can't be any amount of noise. Does that make sense?
Yeah, it makes sense. And do you have any reference for this stuff? Because I think it's can be interesting.
Yeah, of course. So there's this paper by Moore, and in that he references a bunch of other his own papers, papers that have shown that universal computation is possible with let's say neural networks, but all of those are structurally unstable. So if you, I can also send this paper to you if you want. After
no, I took a note. I
Okay,
will
cool.
look it up later.
Of course, yeah. Right. Okay. Okay. So I think the most promising place to search for it is with as at the level of RNA and DNA, and people recognize this going all the way back to the 70s. But most of the previous work have been attempts to build synthetic models of computation using these molecules. And some of these models are universal. But the question that we're interested in is not whether we can build a universal computation system with RNA and DNA, but whether nature has already done so. Okay. But what could it look like? Of course, you can imagine some very complex molecular machine that looks something like a universal Turing machine that operates on DNA. But that sort of machine would have to be huge. Ribosomes, which do the very simple task of translation, are 20 nanometers in diameter, and they're visible in electron microscopy. So a universal Turing machine that would have to follow some much more complex logic would have to be enormous. So the question is, can we imagine a molecular computation system that doesn't require these kinds of extraordinarily complex molecular machines and could have evaded detection until now? And that's what I'm going to try to answer with the model that I'll present. The details of the model that I'm going to present are almost certainly incorrect, but they're only meant as a proof of principle that you can imagine something like this can exist without being undetected. And then we can say, all right, let's try to look for similar systems or some kind of, in the space of all possible computation systems that could have evaded detection. Let's find what's going on in biology. All right. Okay. So the model that I'm going to present to you is all about bridging two fields: RNA molecular biology and combinatory logic, or equivalently lambda calculus, and recognizing the parallels between the structures in these two fields and how they're operated on. Okay. So I'm going to present each one of these. I'm going to first present combinatory logic, what it is, and then briefly how to use RNA biology, or how to imagine RNA biology could be implementing combinatory logic. So in combinatory logic, we have these entities called combinators, and combinators are essentially just functions of functions to functions. They take other combinators as inputs and output a combinator. For example, the I combinator is a function that returns whatever it receives as input. The K combinator can take two inputs and return the first input and deletes the second input. The C combinator takes three inputs, returns the first applied to the third applied to the second. So it basically swaps two of its inputs. So let me just show you how this looks like in action with two simple examples. If I want to compute CKKI, I look up here. What's the rule for C? I have to swap the second and third input. So if I do that here, I get KIK. Now to compute this, to apply the leftmost combinator, I look up what's the rule here. It says KXY equals X. I have to delete the second input. So I just return the first input. So this ends up being I. All right, here's another example with parentheses to show you that you can treat combinations of combinators as a single entity, a single term. Okay, so the really cool thing is, with only a handful of combinators, a handful of these kinds of rules, you can construct a system that's capable of universal computation. So here's a list of a bunch of different primitive combinators and two basic sets, two sets of combinator primitives that achieve universal computation are B, C, K, and W, or S and K. And these are the most popularized examples of universal sets. So with just these four combinators, B, C, W, and K, putting them together in the right way, I can construct functions that solve any computable function. For example, if I want to construct something that solves this transformation of information, something that mimics this Boolean logic circuit, I can do that with this term that I divided across two lines. Okay, so this function, it takes ones and zeros represented with K and Ki, and if I mindlessly apply each one of these rules step by step, I end up with the correct answer that corresponds to the output of this Boolean logic circuit. Now, I want to emphasize that again, Boolean logic circuits are the weakest form of computation. This is just an example of something that combinatory logic can do, but you can also implement more complex functions that Boolean logic circuits can't compute. So, for example, you can have recursion, you can implement unbounded addition, you can basically any program, any algorithm that you can code up in your favorite programming language, you can also code it up in the language of combinatory logic. Okay, so it's a very powerful, versatile computation language. Now, what about how do I implement this with RNA? Well, representing strings is trivial with RNA. We even have a precedent for that in biology. We know that biology uses RNA strands, which are composed of an alphabet of nucleotides, 4 symbols, to represent strings of amino acids. So it's using these codons, using triplets of nucleotides. So it's easy to imagine some kind of representation scheme that maps motifs of nucleotides to the symbols of combinatory logic. But implementing the rules of combinatory logic is where the challenge lies, not just representing. So representing terms is easy. We want to figure out what kind of rules can we imagine happening at the molecular level that correspond to the application rules of combinatory logic. And we know that RNA strands are frequently edited. One of the most common editing rules is splicing. Almost all RNA strands undergo splicing, where some part of the strand is excised and the two ends are ligated together. What I want to do is try to imagine how can we get editing rules like this, very simple operations like this, to map onto a set of application rules that compose a universal computation system. Okay. Now, the most challenging part of doing this is parenthesis matching. Okay, in combinatory logic, if I want to execute the application rules properly, I have to know what a full term is. So if I'm confronted with a open parenthesis symbol, I have to be able to tell where the corresponding close parenthesis is to treat that whole stretch of nucleotides as a single term, to maybe swap it, delete it, or do whatever I want to do with it. So if I show you this string and I ask you what's the corresponding close parenthesis to this open parenthesis, you can't do it immediately. You need to scan the string with your eyes, keep track of the parenthesis depth, adding for every open parenthesis, subtracting for every close parenthesis, until you get to 0. And then you can call this entire stretch of symbols a single term, and then do whatever you want to do with it. Now, to imagine something like this happening at the molecular level in natural, endogenously in cells, that would be too much to expect from an undiscovered molecule, because you'd have to traverse an RNA strand, which already kind of looks like what a ribosome is doing, and you also have to keep track of an arbitrary integer, add, subtract. There's a lot of things that would need to go on to actually carry out this operation that we do in our heads. But fortunately for us, RNA strands already solve this problem of parenthesis matching. The same way that DNA strands can come together and form double helices, RNA strands can also base pair with each other. And sometimes the base pairing happens within the same strand. So it's two different segments of the same strand can come together and form these base pairs. And what results are these highly intricate tree-like structures of base paired segments of the same RNA strand. Now we can interpret these base pairings as parentheses. So if I'm scanning an RNA strand, when I approach one of these stems, I can call this part an open parenthesis. The content inside the stem, which is called the loop, you can call that the content. You can interpret that as the content within the parentheses, and then this other part of the stem would be the closed parenthesis. All right, and this example that I'm showing on the right is an actually existing non-coding RNA strand. So it doesn't code for proteins. It's over 2,000 nucleotides long. It's expressed in skin cells. There's a debate whether it's really important or not, but it's highly conserved. So there's kind of like a lot of mysteries going on with non-protein coding RNAs. Okay, so if we interpret secondary structure as encoding a parenthesis structure, now I can give you examples of what the operation rules could actually look like in the RNA world. So the applications of combinatory logic, I can show you how you can implement them with very simple editing rules in RNA. So here's one example that shows how to apply the B combinator. The rule for the B combinator is shown on the right over here. And one important detail here is that I'm using a different parenthesis convention from what is typical in the field of combinatory logic. In combinatory logic, usually they use left parenthesis convention, meaning that if you write X, Y, Z, that means a parenthesis around X and Y, and then a whole parenthesis around X, Y, and Z. But that makes the difficulties with that. If you want to implement that with RNA, for some reason that I can get into, right associative parentheses suddenly magically kind of work out nicer, so that you only need cleavage and ligation to apply these rules. So okay, so putting that detail aside, this is the application rule for B. Now let's walk through how this transformation here corresponds to this transformation here. First, I'm going to show you how this RNA structure corresponds to this term here. So if we start from the 3' end, you all can see my mouse, right? Yes, okay. So if we start from the 3' end, this part can be interpreted as open parenthesis, then open parenthesis, then open parenthesis. And as I'm reading this, you can notice how this corresponds to the symbols over here. And then the B motif, which could be some hypothetical motif or some codon or something that represents the B combinator. And then we have X, and X could be anything. It could include other stems inside itself, and that's how you can get variables, basically nested parentheses inside parentheses. So that's X, and then close parenthesis, and then Y, and then close parenthesis, and then Z, and then close parenthesis. So X, Y, and Z could be anything. And the structure on the right here also corresponds to this. So start from the 3' end, open, open, X, close, open, Y, close, Z, close. And I just read out this string over here. To get from here to here, all you need to do is a few cuts and ligations. So you only need an enzyme to come in, recognize the B motif, and then perform cleavages and ligations at fixed relative positions to where it found the B motif. And then you can get this structure on the right. And then that enzyme could, float off and do something else. And these are examples. These are the transformations for two other combinators, for the C combinator and the K combinator. And again, all of these can be implemented with only cleavage and ligation, locally defined. Because we have this parenthesis structure, the enzyme that comes in to carry out these application rules doesn't really need to care about detecting what counts as a full term, where the matching parentheses are. It's already there in the RNA structure, and that makes it easy to imagine to already occur. Okay.
Can I ask a quick question?
Of course.
For the strings, there is an intrinsic left to right ordering, right? And I noticed that for the RNA strands, you chose to go from the right strand. Is there something intrinsic about the RNA strands that sort of conveys this right to left ordering, or you just chose to do it that particular way?
Maybe I understood your question. If I didn't, correct me. But RNA strands, they have an intrinsic directionality, and people usually denote that with the 3' end and the 5' end. So there is a directionality in how you can traverse an RNA strand. In fact, that's how a single RNA strand can uniquely encode a protein structure which is a string of amino acids without accidentally coding for the reversed representation. Was that your question, or?
Yeah,
Okay.
that answers the question. Thank you.
Yes. So there's no ambiguity in terms of what the string represents because of directionality issues. Okay, so these three combinators by themselves, they don't form a universal set. So going back to this chart over here, I've shown you B, C, and K, but not W. And the reason I didn't show W is that I can't imagine W happening with only cleavage and ligation, because W requires something more complex. It requires duplicating a strand of arbitrary length. So if you look at the rule for W, it says WXY equals XYY. So there's a term that appears twice on the right side and once on the left side. And to imagine something like that happening naturally in cells is kind of difficult. You need some kind of enzyme to go in and duplicate a strand of arbitrary length and then reintegrate that back into the strand, the template strand. And we have things like RNA-dependent RNA polymerase, which uses an RNA strand as a template to produce another RNA strand. But first of all, we can't imagine a way for that to systematically integrate that back into the same strand. And also, RNA-dependent RNA polymerase doesn't exist across all life forms. And I'm trying to imagine a system that could exist across all life forms and not be detected yet. Fortunately, again, there's another trick that can help us circumvent this need for the W combinator. And to get to that, I want to first introduce how we can implement variables and referencing with this system of computation. So if we interpret parentheses as being represented by these RNA duplexes, I could imagine a system of addressable memory within the cell, where every variable is represented with an address sequence, and it can be referred to by its reference sequence. So a strand that has that's representing a variable might have the address sequence on its 5' end and the content of that variable in the rest of the strand. And then another strand could reference that sequence with the reverse complementary sequence. When these two strands come together, they can base pair, and with only a single cleavage and 2 ligations, you can get the content of that variable nested in parentheses exactly where it was referenced. Okay, so this creates a system where you can have a shared addressable memory space within a single cell, where RNA strands can interact with one another. They can refer to other strands, and they can even refer to copies of themselves. And through that, you can implement recursion. So if we have recursion, we really don't need the W combinator, because one of the uses of a W combinator is to basically define a function in terms of itself. But if we already have a system that's capable of doing that, we don't need the W combinator. So this system is it slightly deviates from combinatory logic because it has explicit variable naming and it has explicit references. And to prove that this is a universal computation system, I had to construct a proof that you can simulate any Turing machine with linear algorithm complexity using that addressing and referencing system and only these three combinators. Okay, so without the W combinator, with that addressable memory system, you can still achieve universal computation. Okay, as an example of what you can compute with the system, I constructed this RNA program that solves unbounded addition. It consists of two functions, this A function and the B function. B is recursive, it refers to itself, and A is recursive, it refers to itself and also refers to B. And to calculate something like 6 plus 2, you can do that through building this strand, which has some part of the strand is representing the number 2, some part of the strand is representing the number 6 in some contrived binary representation scheme, and this strand refers to the program A. So through those rules, A gets integrated into this strand. The operations, the B, C, K operations get applied. They can be applied in any order, so the order doesn't matter. That's another cool thing that kind of magically works out with combinatory logic, where the order of operations doesn't matter in the end result. So it can happen in parallel. And at the end, you get a strand that represents the number 8, which requires 4 digits, the inputs were a 2-digit number, a 3-digit number, but the number 8 is a 4-digit number, but that's fine. It can deal with adding digits. And if I simulate this, just to show that it's efficient in terms of its algorithmic complexity, you can see that the number of operations grow logarithmically with respect to the sum, the final result. Okay. So this is just to demonstrate. I don't think this is how addition actually happens in cells. This is just to demonstrate that you can treat this system as a programming language, just like any other computer programming language. You can construct any algorithm you want. There's no like non-linear slowdowns in terms of the number of operations. So to summarize, DNA contains, to summarize the theoretical model, DNA contains a static library of functions which are used for computation. And sequences which encode these functions are transcribed from DNA to RNA. And a handful of cleavage and ligation rules execute the steps of these functions. I showed you 3 rules, but you could imagine other sets of combinators that execute different kinds of application rules and also reach that level of computation power that's needed for universal computation. And a function can refer to any other function, including the copy of the same function. And this implements recursion. Okay, so this is a theoretical model. And the question is, does something like this happen in cells? Obviously, we don't know. But the idea behind this model was to inspire empirical investigation. So now that we know that something like this can exist and can be mediated through very simple simple rules that could have gone undetected. Now the goal is, okay, can we try to find how nature actually does this? And can we use some of the details in these kinds of models to guide us in finding a universal computation system? So those are the steps that I'm currently pursuing. One of the things I'm looking at is if this is right, if RNA strands are being edited, it should be possible to find traces of these editing rules in sequencing data, if you're using the right kind of method for sequencing. So what I'm looking at first is evidence for swapped segments in RNA strands. We already have ways to imagine how segments within a strand can be deleted with things like splicing. So that wouldn't be surprising. But if we can find systematic rules that determine how two parts of a strand can show up in reversed order, that would be surprising, and that might lead us to maybe one of the operation rules that implement a universal computation system. So what I'm looking at right now are high fidelity full length reads sequenced with PacBio from zebrafinch brain tissue. The data is publicly available. It's published by the Jarvis Lab here at Rockefeller University. And this example that I'm showing you on my screen is one of the biggest examples that I found in this data set. You can see that there are two reads, two RNA strands that contain sequences that are both over 1,000 nucleotides long, but appear in reversed order. And we currently don't know of a canonical rule that could, or a canonical, I don't know, RNA modification rule that could lead to having parts of the same RNA strand swapped. And this is just one example of a hit, but if I plot the distribution of all the sequences that I found, all the identified pairs ordered by the sum length of the swap sequences, I get this histogram over here. And I'm only showing you the far right end of this distribution. On the left side is hidden because it's like there's like a really huge peak, which is where all the noise is. And then there's this distinguished clump of hits. And I think this is one of the reasons why I think this is not just noise and something systematic is happening is you can see this this set of all these hits that are very distinguished from the noise cluster. Okay, and another direction that I intend to pursue is I want to look at learning and memory assays in behaving organisms and try to find correlates of memory in RNA sequences. So most of my postdoctoral work is working on flight path integration, and ultimately I want to find RNA correlates of memory of the spatial location, not just in the expression levels of known transcripts, but in the actual primary sequences of the RNA reads. I'm hoping that we can find unique reads that might represent cognitive memories within these organisms. And I don't want to limit this just to flies. I want to look at other tasks and other organisms and collaborate with other people to try to find RNA correlates of learning and memory in other species. So an obvious question, if RNA is underlying cognition and memory, an obvious question is where is the interface, where's the interface between this RNA system and, like, I don't know, neural spiking? And you can imagine the different possible input and output mediums to this RNA computation system. And there's a recent paper that proposes that the same way that we have nanopore sequencing that convert RNA sequences to membrane potentials, you can imagine maybe something similar like this is happening inside cells, where you can convert precise the precise primary sequence into a sequence of voltage signals. So I want to end with a quote from an underappreciated cell biologist that I believe was far ahead of her time. Beatrice Gelber was studying adaptive learning in the paramecium, a single-celled organism, back in the 1960s. And in one of her papers, she characterized her work as a new approach to behavioral problems, which might be called molecular biophysics. Simply stated, it is the hypothesis that the memory engram must be encoded in macromolecules. Possibly, the biochemical and cellular physiological processes which encode new responses are continuous across the phyla, as genetic codes are, and therefore would be reasonably similar for a protozoan and a mammal. And I really like this quote because one of the recurring themes in the history of science is realizing that phenomena that look very different and appear to be dissimilar are actually governed by the same principles, like the falling of an apple and the orbiting of the moon. And I think that would be a very exciting prospect for the future of biology. And with that, I'd like to thank all the people who have helped and inspired me in this work. Gabby Mayman, my advisor here at Rockefeller, Jeremy Dittman, Abbas Rizvi, Randy Gallistel, and John Mack. And thank you all for listening.
Super, thanks so much. Any questions? Anybody else?
I did have another question. Could you explain a little bit more how the address referencing works? Because in computers, addresses have a sort of convention, an implicit convention. And the other sort of concern is that, at least coming from something like C programming, the address variables are explicitly defined in a way that's different from other variables. So they use the star convention. So star variable means it's a referencing variable. So when it comes to RNA strands, are there like specific differences between strands that enable or facilitate this sort of distinction between memory variables and ordinary variables?
Yeah, that's an excellent question. And that is a detail that I kind of glossed over. So whatever the representation scheme is, it cannot interfere with how the combinators are represented. So ideally, there wouldn't be any ambiguity in when you're looking at a sequence whether this is whether this is like referring to another strand or whether it's representing, like, I don't know, B applied to C applied to K. And you could do that by coming up with unambiguous representation schemes for the same way that RNA strands represent amino acids. They also need to represent operations like start and stop for the ribosome to know when to stop when to stop translating and when to start. And it uses it uses the same scheme of representation. It uses codons. So you need some way, but it doesn't like what is it called? It's not ambiguous. So it's not like sometimes it's not clear whether this is a stop codon or a certain amino acid. So you just have to come up with a representation scheme where that differentiates between motifs that are used for address sequences or the combinators. Does that make sense? Like for example, I don't know, it's going to sound very arbitrary. That's why I didn't really give an example. But let's say if it starts with GC, then the following, I don't know, 10 terms are going to be the following 10 nucleotides are going to be an address. But if it starts with I don't know, AC, sorry, if it starts with like AC, then it's going to be one of the 3 combinators, something like that.
Yeah, that makes sense. How about the address convention? So, for example, if we have like a single-dimensional grid, then you can say that the leftmost location is 1, and then as you go on to the right, it's 2, 3, 4, and it's like a 2-dimensional grid. Then we can assign some sort of convention, but it sort of suggests that there must be an agreed-upon convention and it's stored somewhere. How does that convention work here? Is there sort of an implicit convention of some sort?
Yeah. So the space of variables is not really. It doesn't. There's no way to spatially organize it necessarily. There's no single way to spatially organize it. It would just be every sequence would correspond to a variable. So it's kind of like the space of all strings. As if you have as if every string would represent a different variable, and you can refer to that string. And these RNA strands are kind of floating around in the cell. So an RNA with a certain address could be anywhere in the cell, or I don't know. Those are details which are kind of even more speculative than what I've speculated on already.
I have a question about something that I mentioned before. You mentioned that I truly understand that some devices, for example, vacuum cleaner, don't really need a universal machine to operate. They can do whatever they need to do with some analog computation. Maybe we don't need also the universal computers in order to describe consciousness or whatever we'd like to describe. Maybe we created a machine that have more complex theoretical complexity, mathematical, more than us, but they are not necessarily needed to be in order to describe us. That's make any sense? So maybe I'm saying that the whole search for your universal Turing machine in nature is maybe not the right question.
Yeah, that absolutely makes sense, and that's how I used to think about it. But my thinking has kind of changed for several reasons. One is that I might be repeating myself, but if I am, feel free to interrupt and say what your concern is about these arguments. So first of all, biology could come up with very complex systems through evolution that abide to the principles of optics. Like our eye has an aperture, it has a lens, adjustable aperture, and it looks really much like a camera. And it's not just a coincidence. They're following principles of optics. If you want to project a 2D image of your environment, you kind of have to follow these rules. And then for universal computation, I feel like it's even less complex. It's even easier to accidentally stumble upon a universal computation system. Like it's hard, like my intuition is that you need Darwinian evolution to evolve an eye, but you don't necessarily need Darwinian evolution to evolve a universal computer. You might just accidentally create it by chance without a Darwinian evolution or selection. These two things kind of make me think that it's very likely for it to already exist and then be used for domains that don't necessarily need it. But it's also possible, and I have to acknowledge this. It's possible that maybe it just doesn't exist in biology. Maybe as you said, universal computation is a man-made construction. But I guess there's just something about that just doesn't sit well with me. Like
I completely agree, but I'm saying maybe universal Turing machine is machine that is very, very sensitive in terms of input and output, is not like analog computers, which analog computers are much more robust in terms of definition of all the input range and output range, and you can decompose and compose more parts on that, and which universal Turing machine, you require a lot of definition and a lot of interface in order to define exactly the input output of each one of those machines. So maybe that's why biology is not doing this.
I don't know if my intuition agrees with that. Like I feel like my intuition is that digital computation is much more robust to noise, and especially because it can deal with issues of noise at the algorithmic level.
Right.
Like you can have checksums and stuff like that, and the nature of a digital computer, there's like discrete attractor points for bits and things like that. I don't see the advantage of analog computers, especially in terms of robustness to noise. But I
agree in terms. I agree in terms of complete noise. Mm-hmm. But in neurons, for example, there are sometimes spontaneously spikes. They create a lot of noise inside the mechanism itself.
But that's an assumption, right? Like we don't know what those spontaneous spikes are. They could be, they could mean something, and we just haven't discovered what the code is, or especially, I mean, it doesn't contradict this kind of model to see spikes that we can't explain.
Right. Yeah, I understand your point. Yeah.
Okay. It's not a question. I just want to hear what you think about it. Unless you're not done, Hannah, now. So you can ask. I don't know if I have seen anyone directly say this, but I could be very wrong because I'm bad at reading. So often when we search for universal computing systems, you take a system like this RNA idea and say like, this could do universal computation super fine. Could it also be though, or like if we go back to the Moore paper, you have a dynamical system that does not have structural stability, so this is hard, blah, blah, blah. But it could be that you have many different subsystems that then in and of themselves as an individual system are not that, but when coupled together, they could be. So could you build a universal computer? It's out of pieces that are not universal computers. Like you put neural activity on some of these at some point. Okay, so like maybe neurons in and of themselves are not universal computers, nor are LFP fluctuations, right? But when you couple the two things together, when you combine them, they can then do universal computation. So instead of looking for one individual system that can be this thing, maybe it's wiser, or has anyone thought about this, or have you thought about this? Could it be that you only find this when you couple things together? Like, a tape doesn't have to be a tape. What if the tape is this and the head is this and then the computation? So if you couple things together, it might be that like system A, when reading from systems B and C, is all of a sudden universal in a given context, but in another context, it's not, because it needs to decouple. So I imagine, in my head at least, that you have multiple dynamical systems coupling and uncoupling from something, that when they're coupled a certain way, they're universal. When they're not, they're not. And what do you think about that? Is that insane?
Yeah, I totally get it. I think so. I don't know of an example of this where, like, you have, I don't know, you can put LFPs and something else together and then suddenly becomes universal. But I think tangential to your point, you could use neurons to construct a universal computation system. It just wouldn't be a traditional neural network. So for example, to give you an example of how it could work, you could simulate cellular automaton, a cellular automaton system with neurons. And people have come up with models for that. And if you have the right update rules for your cellular automaton, you could achieve universal computation, like Conway's Game of Life. So it's not that like neurons in themselves they can't reach this level. It's just you have to explain what the system is. Like the difference between that system and what we conventionally think of as neural networks is with a cellular automaton simulating model. When I say my network solves addition or solves this problem, what I mean by my network is like an initial activity pattern. I'd be saying, okay, this on, this on, this on, this off, this off, this off. This is the solution to addition, and this can be embedded in a neural substrate of arbitrary size or unbounded size or whatever. And if it runs out of size, you can embed it in a larger network, and then it'll compute the problem that you want. But when people say neural networks, what they typically mean when they say, hey, this neural network solves this problem, is nothing like that. What they mean is a set of precise weights, the actual nodes, and like the activation functions that to like put them together, create a neural network. That's the thing that solves an input-output function, and that's what I'm criticizing here. That doesn't, as far as we know, that doesn't arrive at the level of universal computation. But it's very possible to imagine it happening, I don't know, with neurons, or even at the cellular level, like implement cellular automaton using just single cells that aren't neurons. It's just